Hey guys, how's it going? So I was looking through your guys' work and it looks like you guys were needing some help with the dihybrid crosses. So I decided to do a little example like we did in class. The current Zoom setup I have doesn't really allow for me to like rearrange while we're doing the Zoom meeting. So I decided to do a little video for you guys. So I'm using the example problem that we had in our actual lecture. So when you guys had a little lecture, I think in week 2, that explained how to do these dihybrid crosses. So I'm using that example. So it looks like this. And it's the very same example problem as your guys' lecture. So it says, in pea plants, purple flowers, capitalized P, is dominant to white, and tall plants, T, is dominant to short plants. So just like when we were learning monohybrid crosses with just the straight dominant and recessive features, anytime that you have a capitalized letter, it's always going to dominate that trait, and that's what's going to be expressed. So we're going to make a key just like when we did in class. So trait number 1 is color. So dihybrid is just that it's going to have 2 traits that you're looking at it. So the first one is color, and it's telling us that P is purple. That is dominant to white. Sorry, you can hear my silky D puppins. The second trait it says is that capitalized T are for tall plants and they're dominant to short plants, which is the lowercase t. So that means that the second trait is going to be for height. So trait number 2 is for height. So T equals tall, and the lowercase is short. And so in the problem, it's going to tell us what to cross. So it says one parent is heterozygous for flower color and short. So then it doesn't say which parent is which, so we're just going to arbitrarily put one in here. So one parent is heterozygous for, so we'll make the female heterozygous for flower color. So then she is going to be little p, and then she is going to be, let's see, purple and short. So in order for her to be short, she has to have 2 recessive t's, small lowercase t's, to be short. So she will be T. And then it says the other parent is heterozygous for flower color and heterozygous for height. So dad is going to be heterozygous for flower color and heterozygous for height. And then it says label both genotypes and phenotypes of all the offspring. So it's you guys had some trouble with labeling the genotypes and phenotypes. I don't know if it was that you guys just forgot to do that part or if you just were having trouble figuring that out. So here we go. So now we have the parents. So we have to figure out the gametes that go on the outside of the boxes so that when you put the gametes together, they make that offspring. So this is where the dihybrid crosses are a little bit different than the monohybrid crosses. So in this one, you have to use what's called the FOIL method. So we're borrowing a distribution method by the math world and using it in these Punnett squares. So because actually Mr. Punnett, I think, that created these or came up with the Punnett squares was a mathematician. So here we go. In the FOIL method, you go first to first. So the first set of gametes will be capital P, lowercase t. You go outside to outside. And then inside to inside. And last to last. do the same thing for the dad. So for the dad, first to first. Outside to outside. Inside to inside. And last to last. Try to be consistent for you guys. Sweet. So now that we have the gametes on the outside of the square, now we can put them together and create the offspring. So we'll put the offspring together. You do want to put your P's back together and then your T's back together so that you can figure out the actual phenotype of each of these offspring. So put the P's back together and the T's back together. So we do that for all of them. B, little b. Sweet, so now we have all of the genotypes for the offspring. The genotypes, again, are going to be the different letters that represent the traits themselves. And then the phenotypes are going to be the description, the physical description of these traits. So we've done our genotypes, so now we need to do the phenotypes of each one. So for the first one, it is showing us that there is 2 capital P's. So those P's are going to be representative, again, of the trait color. And since we're not doing any sort of incomplete dominance or codominance, this is just straight dominance and recessive, we know that this one is going to be purple because of its capital P's. And then for the trait of height, it has a capital T, which is going to dominate the lowercase recessive t. So this individual is going to be tall. Same thing. This individual right here has 2 lowercase t's, so just like the mother, 2 lowercase t's would make the recessive traits expressed. So in this individual, this plant is going to be short. capital and tall for this one because it is heterozygous with that capital T. This individual here has two lowercase p's, so because it has two lowercase p's, that would show the expression of white color. So this one is going to be white in color, and it will be tall because of that capital T that it has there. That is going to be dominant over the recessive lowercase t, so this individual will be tall and white. And then these last two here, we have two lowercase p's put together, so that would make that express the white trait for the white color for the color trait. And then it is going to have two lowercase t's, so it will be a short plant. So it was going to be a white plant that is short in height. All right, and that is a dihybrid cross. So hopefully this helps you guys understand how to do these a little bit better. I'm sorry that, you know, we weren't able to do this in class, but hopefully this helps you get a better grasp on how to do a dihybrid cross and also label both the genotypes and the phenotypes. And that was our kitty, Lucy. So Lucy's a star now. I'll see you guys later. Bye.